Dear 12th class students, and today we are going to discuss the types of RNA and the process of transcription. Firstly, we will take up the transcription in bacteria and then in the eukaryotes. And in bacteria, there are three major types of RNAs: mRNA, that is the messenger RNA, tRNA, that is the transfer RNA, and rRNA, that is the ribosomal RNA. And all three RNAs are needed to synthesize a protein in a cell. The messenger RNA provides the template, transfer RNA brings amino acids and reads the genetic code, and the rRNA plays structural and catalytic role during translation. That means the work for the different types of RNA is divided. Messenger RNA acts as a template, transfer RNA brings amino acids and reads the genetic code, whereas the ribosomal RNA, that is the rRNA, it plays the catalytic role during the process of translation. And there is a single structural and catalytic role. There is a single DNA dependent DNA polymerase that catalyzes transcription of all types of RNA in bacteria. Only one enzyme is responsible to catalyze transcription of all types of RNA in bacteria. That is the DNA dependent RNA polymerase. RNA polymerase binds to promoter and initiates transcription, and this process is the initiation. It uses nucleoside triphosphates as substrate. Here you can see RNA polymerase. It binds to the promoter site and initiates transcription, and then here this transcript is elongated, and finally it gets terminated and the RNA is formed. That means it uses nucleoside triphosphates as substrate and polymerizes in a template dependent fashion following the rule of complementarity. It somehow also facilitates opening of the helix, that is the DNA dependent RNA polymerase. It is a multifunctional enzyme and it facilitates the opening of the helix and continuous elongation. Only a short stretch of the RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, the nascent RNA falls off. So also the RNA polymerase, and this results in termination of the transcription. That means finally the formation of the transcript is complete. Now the question arises here: the RNA polymerase is able to catalyze all three steps, which are initiation, elongation, and the termination. The RNA polymerase is only capable of catalyzing the process of elongation. It associates transiently with initiation factor rho and the termination factor. To initiate and terminate the transcription, respectively, association with these factors alter the specificity of the RNA polymerase to either initiate or terminate the transcription process. And in bacteria, since the messenger RNA does not require any processing to become active, and also since transcription and the translation takes place in the same compartment, there is no separation of cytosol and nucleus in bacteria. Many times, the translation can begin much before the messenger RNA is fully transcribed. And consequently, the transcription and translation can be coupled in bacteria because of because these processes occur in the same compartment. And in eukaryotes, there are two additional complexities. There are at least three RNA polymerases in the nucleus, in addition to the RNA polymerase found in the organelles. And hence, in eukaryotes, there is a clear-cut division of labor. That means all these polymerases have definite function to perform. The RNA polymerase 1 transcribes ribosomal RNAs, 28S, 18S, and 5.8S, whereas the RNA polymerase 3 is responsible for transcription of transfer RNA, that is tRNA, 5S rRNAs, and snRNAs, that is small nuclear RNAs. The RNA polymerase 2 transcribes precursor of mRNA, the heterogeneous nuclear RNA, that is hnrRNA. And the second complexity is that the primary transcripts contain both the exons and the introns and are non-functional. Introns are non-functional. Hence, it is subject to a process called splicing, where the introns are removed and exons are joined in a defined order. Heteronuclear RNA undergoes additional processing called as capping and tailing. In capping, an unusual nucleoside, that is methyl guanosine triphosphate, is added to the 5' end of heteronuclear RNA, and in tailing, adenylate residues 200, 300 are added at 3' end in a template independent manner. It is the fully processed hnRNA, now called messenger RNA, that is transported out of the nucleus for translation. And the significance of such complexities is now beginning to be understood. The split gene arrangement represents probably an ancient feature of the genome, and the presence of introns is reminiscent of antiquity, and the process of splicing represents the dominance of RNA world. And in recent times, the understanding of the RNA and the RNA-dependent processes in the living systems have assumed more importance, and this clearly shows that the primitive genetic or the hereditary material was the RNA, and now taken over by the DNA in higher organisms. Thanks. Have a nice day.